Hello, dear students, welcome back to Plus 2 Biology. Biotechnology, its applications. So, applications of biotechnology in agriculture, we had a look, and today we are discussing applications of biotechnology in medicine. Okay, and we are just briefing the major applications of biotechnology in medicine. Okay, the three main areas where biotechnology has its application in medicine are in the production of therapeutic drugs, in the gene therapy, treatment of the genes, and also in the disease diagnosis. So, biotechnology has its major applications in the production of therapeutic drugs, in gene therapy, and also in disease diagnosis, early diagnosis of the diseases. Okay, let us discuss one by one. The first one, the production of therapeutic drugs using the genetic engineering technique. So, this is a major branch of concern and many researches are going on in this field to produce medicinally useful drugs using bacterium. By the gene transfer, the gene for the particular medicine or the drug is transferred to the bacterium and is multiplied, and the drug is produced in bacterium using bacterium. Okay, likewise we have produced a number of therapeutic drugs. Okay, about 30 are now very common in account and 12 are being heavily marketed in India. Okay, so about the therapeutic drugs, when we discuss as our example, every time we use the case of insulin. Here also we can just discuss the case of insulin. Okay, you know the fact, the production of insulin, under the therapeutic drug production, the production of insulin we discuss as an example. Okay, scientists face a great trouble in producing insulin. You know why? So, just understand, insulin is produced in human body as proinsulin. That means, when scientists isolated insulin gene from human body, they were not able to produce insulin, but when it is expressed in bacterium, it produced proinsulin, not insulin. So, understand, when insulin gene expresses in our body, it produces what? Proinsulin, not the insulin directly. Under the conditions of pancreas, within our body, the proinsulin is matured to insulin. So what happens here? You know, proinsulin has 3 polypeptide chains. A peptide, B peptide and C peptide chains. The A peptide and B peptide chains are connected through a C peptide chain and also through disulfide bonds. Okay, whereas when this proinsulin is matured to insulin, insulin has only 2 polypeptide chains, the A chain and B chain. C chain is absent in the matured insulin, it is removed during maturation. So, when proinsulin is matured to insulin, what happens? The C peptide is removed. So that the insulin has only the 2 peptide connected by disulfide bonds. Okay, so this problem faced by the scientists in order to produce insulin from insulin gene encoding was when insulin gene is encoded to bacterium, Escherichia coli bacterium, it produced proinsulin, not insulin. Okay. Just understand, the insulin gene as such when is taken, it is forming proinsulin and it should be matured inside the body to form insulin. So, it will take time for the maturation of proinsulin to insulin for a diabetic patient. So, it is mandatory to supplement insulin other than proinsulin. So, what can be done? Scientists thought about this and they found that in the insulin gene, there are 3 separate regions coding for, gene A, B, C, coding for A peptide, B peptide and C peptide. So, it was an American company, Lilly in 1983 successfully produced the insulin by encoding the gene for A peptide and gene for B peptide separately in E. coli. Here just understand trouble once again. Human insulin gene as such will be producing proinsulin instead of insulin, and during maturation of proinsulin to insulin in our body, the C peptide is removed and the real insulin has only what? A and B peptide. So what the American company Eli Lilly has done is they have taken a gene for A peptide and then another gene for B peptide separately grown or encoded this in E. coli, and they produced, instead of producing insulin, they obtained A peptide and B peptide, not the insulin as such. Then this different number of A peptides they obtained, number of B peptides they obtained, then artificially they joined this by disulfide bond. Okay, so these two peptides were joined artificially by disulfide bond to produce insulin. So it was a success in 1983 what this company did. Okay, so this is about the production of what? Genetically engineered insulin, the problem faced and how is solved. Okay, before the genetically engineered insulin production, it's known as Humulin. The genetically engineered insulin, human insulin is known as Humulin. Before the production of Humulin, the insulin was obtained from cattle, especially from pigs and all. At the time of slaughtering, from their pancreas, the insulin was taken and is injected to or introduced to patients. This made a lot of allergy and mental problems to the patients when they came to know that, the source. Okay, now this is an alternative method, and genetically engineered insulin is nowadays highly marketed. Okay, then we can move to the next session, the application of biotechnology in the gene therapy. What do you mean by gene therapy? What are genetic disorders? We have discussed already. Genetic disorders are disorders due to a defect in the gene. Okay, so what is gene therapy? The replacement of a defective gene by a normal gene is called actually gene therapy. Okay, the clear example given in a textbook is ADA gene therapy. Okay, ADA gene is essential in our body. It is to produce ADA, adenosine deaminase enzyme. So, ADA, the gene in our body is responsible for producing the enzyme adenosine deaminase. Okay. So, about this enzyme, what is the role of this enzyme in the body and what happens in the absence of this enzyme? This enzyme has a very crucial role in the maturation and working of lymphocytes. For the lymphocytes action, this enzyme is very significant in our body. So, an absence of ADA enzyme will affect our lymphocytes. That means it affects our immunity. We may be subjected to other pathogenic diseases. It is called severe combined immunodeficiencies. SCID it is called. Anyway, ADA gene therapy means replacement of defective ADA gene by normal gene. A patient with ADA disorder, I mean, if the gene is defective, we can substitute normal gene to that patient. Okay, so what is done in ADA therapy? It was in 1990 as a first case of clinical gene therapy. It was carried out to a 4-year-old girl, ADA gene therapy. What has done at that time? Okay, in the ADA gene therapy, at first, lymphocytes were taken. I mean, from blood, lymphocytes were taken out from the patient's body. Okay, so lymphocytes from the patient's blood are grown and taken out and grown in a culture medium. Okay, that was the first step. Secondly, you know that lymphocytes are having a defective ADA gene. Okay, so what has done as a second step is replacement of that gene. Actually, the gene is not cut and taken out and removed. Functional ADA cDNA is introduced to the lymphocytes of the patient using which as the vector, retrovirus as the vector. So with the help of retroviral vector, functional ADN gene, ADA complementary DNA is introduced to all lymphocytes of the patient. Then now the lymphocytes genetically engineered, these lymphocytes are, and these are normal, and these lymphocytes are returned back to patient's blood. Okay, and you know this particular lymphocytes having a lifespan. After that lifespan, the genetically engineered lymphocytes die. Again, patient go back to the normal state. So what we should do? We should do the periodic infusion of genetically engineered lymphocytes. After the lifespan of lymphocytes, when it die, the patient should be again infused with the genetically engineered lymphocytes. Okay, so once again, in the ADA gene therapy, ADA gene is essential for the production of adenosine deaminase enzyme, which is very significant for the action of lymphocytes. Okay, and it affects our immunity and lead to skin-like problems later. What is done in the first case of clinical gene therapy in 1990? The first step, they have taken out the lymphocytes from the blood of the patient, and then they have cultured it in large number. Then these lymphocytes were treated with functional ADA gene. That means ADA cDNA is introduced. They were introduced with the help of retroviral vectors. Okay. Then after that, this cultured, processed lymphocytes, genetically engineered lymphocytes were infused to the patient's blood. Okay. Then it was successful, and this lymphocytes will die after its lifespan. So it required periodic infusion. That was the only problem. Okay. Then, and now you know that if it's needed as a permanent cure, what we can do? It can be a permanent cure if this gene therapy is done in early embryonic stage in the stem cells. The stem cells, these embryonic stem cells are forming lymphocytes. Okay, the stem cells, if is treated with this normal ADA gene, this will produce always the normal lymphocytes. So it could have been a permanent cure. In order to make this as a permanent cure, to avoid the periodic infusion, what can be done is do the gene therapy in the early embryonic stem cells. Okay, that's all about the gene therapy case. Example, ADA gene therapy. Okay, we have one more small topic left under application of biotechnology in medicine. The third part, that is disease diagnosis. Disease diagnosis. Okay. How we diagnose our disease? Whenever we feel any discomfort or any symptoms like fever, headache, stomach pain, or like any symptoms. When the symptoms appear, we go for diagnosis, right? So that means that time the disease pathogen will be plenty of in our body. It might have multiplied in our body. Then only we get the symptoms. Whereas some dreaded diseases like cancer, HIV, etc., we can diagnose early with the help of genetic engineering technique. Our usual diagnosis technique include blood test, urine test, serum test, and also in the internal diseases we do scanning like things. But in the case of early diagnosis, we have three main strategies, three main methods for disease diagnosis. So disease diagnosis can be very early done using the biotechnology or genetic engineering. It is helpful for the dreaded diseases like HIV, cancer, etc. Okay, then such pathogens, oncogenic viruses and HIV can be detected very early by PCR technique, DNA hybridization technique, and ELISA. Okay, the three major techniques used in early disease diagnosis, which has, the biotechnology has its application, are PCR, DNA hybridization, and ELISA. Okay, these two you are familiar. PCR, what is it? Even if it's one pathogen in our blood or body, it can be amplified. The pathogenic DNA or RNA can be amplified and we can have an early detection. Okay, I mean if it's 1 or 2 in number, we may fail to recognize it, but it can be very easily identified after amplifying its genetic material. So that is done in PCR, amplification of the genetic material of the pathogen and early diagnosis, even in very trace amount. Similarly, in DNA hybridization, what is done? We have ready-made probes for all antigenic DNA, and with the help of that ready-made non-probes, we will hybridize the pathogenic DNA and identify it very early. Okay, for HIV we have this particular probe, means if HIV is present, it will go and bind and it will be autoradiography will reveal the presence. Okay, so with the help of probes, we can identify the disease pathogen very early by hybridization and autoradiography. This is called the DNA hybridization. Whereas the third step, it may be new for you. It's very popular as HIV test, ELISA. And what is done in ELISA is, it is based on antigen-antibody interaction. Okay, the ELISA test is based on antigen-antibody interaction. That is the principle of ELISA test. Okay, what is done here? Suppose if in the test, okay, the patient's blood serum is taken and patient's antigen. Okay, this is patient's antigen. We don't know what type of thing is this. Okay, patient's antigen. Okay, then it is we will give number of ready-made antibodies. Antibodies are specific, and these antibodies go and bind to this antigen, and we will be coupling this antibody with enzymes. That's why it is called enzyme linked. Okay, so wherever this action is, we will get the color. Okay, in order to get that easily, make it more sensitive, we are attaching it with enzyme. So the name is enzyme linked. Okay, so we are recognizing the patient's antigen with the help of ready-made antibodies attached with the enzyme. That is what is the technique ELISA is. It is based on antigen-antibody interaction. Details are not there for you. Just understand, ELISA is an early diagnosis technique which is working based on the principle antigen-antibody interaction. Either the patient's antigen or the patient's blood serum antibodies are detected using the respective antibodies or antigens. Okay, based on the coagulation, based on the interaction and based on the enzyme action, we can very easily diagnose how strong the disease pathogens are in our body. What's the quantity and which type of pathogen it is, we can very easily detect. Okay, this is what about the application of biotechnology in medicine. 3 main areas: the production of insulin, then the therapeutic side, second gene therapy, ADA gene therapy, and third disease diagnosis. Its elaboration we do in the live session. Thank you.